So I'd like to start by thanking Michael Kennedy and Peter Kopp and their staffs, particularly Anna and Lauren, who've done an outstanding job of taking care of me and organizing this. They really put a lot of work into it. I'd like to thank you for your interest in this topic and for making time to come and listen to this, because one of the things that happens when science gets politicized is that truth sometimes gets lost in the balance, and claims on both sides can be misleading. And in both the federal debate that has occurred over stem cell research, as well as the debates in various states, including Illinois, there sometimes have been claims on the pro side that have exaggerated the potential benefits, and often misleading claims on the counter side as well, to try to muddy the waters in trying to reduce support for stem cell research. So what I want to do today is talk both about adult stem cell research and some of the cool things that we've learned as a result of studying adult stem cells, as well as embryonic stem cell research. So I'm going to try to tell you three stories today. One is about stem cells and cancer. Second is about stem cells and aging. And the last third of the talk will be about embryonic stem cells and what scientists are really proposing to do, and what some of the misleading information is that you've probably heard. Finally, and I want to note this explicitly, there's going to be time for questions at the end. And I think this is important because a lot of people end up spending a lot of time reading newspaper articles about stem cells and come to these kinds of fora with lots of questions. And oftentimes that's the part of these kinds of opportunities that can be most valuable to people. So I do want to make sure we make good use of it. So I'm going to try to be disciplined about finishing in time to leave time for questions, and I hope that you won't be shy about asking them. So let's see. So academic scientists are sometimes, like this guy, accused of living in an ivory tower. The caption here is "Two barbarians and a professor of barbarian studies." But I have to say that living in the world of stem cell biology, I feel like we're living in the trenches. Because not only are these public policy debates going on that people like me, spend a lot of time federally and in the state talking about these issues, and I appreciate the opportunity to talk to you guys, in part because it's an opportunity to really explain what the scientists really want to do and what's really at stake, and what kinds of issues are in the balance. But also because invariably, whether I'm testifying before Congress or whether I'm in a situation like this, people are here who have juvenile diabetes, who have spinal cord injuries, who have other kinds of diseases, who believe that stem cell research represents their best hope. Now, in some cases, it may be their best hope, and in other cases, maybe it's not their best hope. But the fact that so many people, you can't stand in my shoes without feeling a sense of urgency about these things, because every week I get emails from people saying, my husband was diagnosed with this. The doctors say there's nothing we can do. Do you have any clinical trials that you're planning at the University of Michigan or elsewhere? Is there anything you can do? The people that do the kind of work that people in my laboratory do, what we would like to do more than anything else in the whole world is cure something that couldn't be cured otherwise. And people in my lab are working around the clock trying to discover new cures. But the stuff that was easy to cure has been cured. And the stuff that we continue to work on is really hard to cure. And there are many different problems out there, and we're early in this enterprise. So the exciting thing about stem cell research is that it creates opportunities that don't exist otherwise. And I'm going to try to illustrate some of those things that have emerged from our work in this area. But we owe it to those people, I think, no matter what the probability is. Maybe in some cases there's a 1% chance of success. Maybe in other cases there's a 10% chance. Maybe in other cases there's a 50% chance. But whether it's a remote chance or whether it's likely that we're successful, and no matter how people disagree about that, the fact is that we have to do everything we can to do more for those who are suffering and for whom we can't cure them now. So there are many different kinds of stem cells. That's the first thing that you have to understand. There are embryonic stem cells. See, now I don't know how all these buttons work. I think these slides are randomly moving, and I can't tell whether the computer's just getting bored and I'm spending too much time on each slide. So there are many different kinds of stem cells. There are embryonic stem cells, which only exist at the earliest stages of embryonic development, but which have the capacity to make every cell type in the body. Even though these cells in vivo only exist at the earliest stages of embryonic development, literally within days of fertilization, we can propagate them in culture as lines. So when you hear about the NIH-approved lines or the lines that are approved for federal funding, what they're talking about, on the president's approved list, there are 21 embryonic stem cell lines that were created prior to September 2001. Those lines arise from 21 different embryos. So that's the extent of the genetic diversity that those lines represent. So that is one embryo, one line. So once we create lines, those lines can be propagated indefinitely. And so we can continue to use them over and over again for experiments. There are also tissue-specific stem cells, which in the newspaper are often referred to as adult stem cells. Now, adult is a bit of a misnomer because these tissue-specific stem cells exist both in fetal tissues as well as in adult tissues. But there are many different types of these tissue-specific stem cells. There are blood-forming stem cells that exist in blood-forming tissues throughout life. So all of you have blood-forming stem cells in your bone marrow that make new blood cells every day. And you need those stem cells to make new blood cells. And if those stem cells suddenly went away, you would die within weeks of hematopoietic failure. You would be immunocompromised. You wouldn't be able to fight infections. You wouldn't be able to clot your blood, and you'd die of bleeding and infection. You have stem cells in your nervous system, particularly in your brain. I'll talk a little bit later about how there's a population of stem cells in the forebrain that makes new brain cells every day, including new neurons. There are stem cells in your muscles that are responsible for regenerating muscles when your muscles need to regenerate. Stem cells in various epithelia, such as in the wall of the gut, in breast, in skin, in all kinds of places. So there are many different kinds of adult stem cells. And the thing that distinguishes these adult stem cells from these embryonic stem cells is whereas the embryonic stem cells can make every cell type in your body, the adult stem cells are already partially specialized. So blood-forming stem cells can make all types of blood cells, but they can't make new cells in your brain or your liver or your muscle. You need to have those other types of stem cells to make different cell types in different tissues. So why study stem cells? Well, we have lots of reasons. Stem cell biology encompasses fundamental scientific questions at the forefront of cell and developmental biology, questions that have the potential to change the future of medicine. Stem cells offer the opportunity to better understand the origin of disease, because whether you're thinking about cancer or birth defects, these diseases often involve things that go wrong either in the stem cells or in the mechanisms that normally regulate stem cell function. We have the opportunity to perform cellular therapies. That's the tip of the iceberg. So you often hear about Parkinson's disease and juvenile diabetes, where the idea is maybe we could cure these diseases by replacing the cells that go missing in the disease. I'll talk more about that later. But the kind of application that's probably even more important than that, which you hear less about, is in terms of drug therapy, where we may have an opportunity to change the way we discover drugs by using stem cells, by screening new drug candidates against embryonic stem cells. So when President Reagan's family talks about how important embryonic stem cell research is for the future of Alzheimer's disease, that's what they're talking about, the opportunity to discover new drugs that may be more effective in treating Alzheimer's disease. Now, of course, with all of these reasons, this charges up scientists to answer questions that couldn't be answered before about clinicians to try to treat people differently, but it doesn't necessarily charge up the politicians to support expanded embryonic stem cell research. And in this cartoon, it shows two senators coming down the steps of Congress, and one of them is saying, of course, it would be a different story entirely if we could extract crude oil from stem cells. So improving the way we treat cancer biology. I'm going to tell you a story about cancer because one of the main interests in my laboratory is to understand the mechanisms by which stem cells renew themselves. So stem cells persist throughout adult life in a variety of different tissues. In blood-forming tissues, you have stem cells when you're born, and they persist all the way throughout adult life. In your skin, in your brain, in your muscles, in all of these places. And the way in which stem cells persist throughout life is by undergoing what's called self-renewing divisions. That is, stem cells divide to make more stem cells and thereby perpetuate themselves. And in that way, they're different from most cells in the body, which are not able to perpetuate themselves. So it turns out that the process by which stem cells undergo these self-renewing divisions is hijacked by cancer. You hear about mutations or genetic defects that cause cancer. Invariably, these genetic defects inappropriately activate the mechanisms that normal stem cells use to self-renew. And so in that sense, cancer can be considered a disease of dysregulated self-renewal, where stem cells normally self-renew in a highly regulated manner, with some genes that promote the process, other genes that inhibit the process, and that are present in dynamic equilibrium. That equilibrium gets out of balance in cancer, where the accelerator gets stuck on and the cells self-renew out of control. And so in my laboratory, when we study the mechanisms that stem cells use to self-renew, we invariably not only learn something about normal development and how we can enhance regeneration in the tissue, but also how cancer cells get carried away and hijack this process. And thereby, we identify new ways of trying to intervene therapeutically in cancer. So one of the things that we and others have learned in recent years is changing the way that we think about cancer. For a very long time, we've recognized that tumors are packed with very different kinds of cells. Not every cancer cell looks the same. But the idea was, despite this heterogeneity, that many different cancer cells had the capacity to proliferate extensively and form new tumors. Because it was believed that even a single cancer cell could kill you, the modern therapies that we have for cancer involve eradicating every cancer cell. So our first choice is to chop out the tumor if we possibly can. That's a pretty successful therapy. Our second choice, once we have disseminated metastatic disease, is to use these broadly toxic chemotherapies with the hope that we'll kill the cancer before we kill the patient. But of course, these approaches are so toxic that oftentimes we do kill the patient before we're able to cure the cancer. In contrast, in a number of important cancers, including leukemias and breast cancer and colon cancer and various brain cancers, a very different model of cancer has emerged in recent years called the cancer stem cell model, where the idea is that in a normal tissue, we have a few stem cells that have the capacity to proliferate extensively, and a large number of other cells that arise from the stem cells and that have a very limited ability to divide. So most of the cells in your blood, they arise from rare blood-forming stem cells in your bone marrow, but once they've matured in your blood, they have a limited ability to proliferate. The same thing turns out to be true in cancer, that cancers, at least some types of cancer, are driven by infrequent cancer stem cells, which retain the capacity to proliferate extensively and to form new tumors. But just as a normal stem cell gives rise to diverse types of specialized cells with a limited ability to proliferate, so do cancer stem cells, giving rise to large numbers of cancer cells that have a limited ability to divide. So, in fact, in testicular cancer, this is the one solid malignancy that we can cure pretty effectively even after it becomes metastatic. We've known for about 30 or 40 years that there are rare testicular cancer cells that are undifferentiated, and those are the ones that we really have to kill. And they give rise to all kinds of crazy other cells, hair and teeth and neurons and crazy stuff that loses its ability to divide in becoming specialized. It turns out that that's also true of other cancers like the ones that I've listed on here. And so if that's true, what this really means is that rather than using broadly toxic approaches to try to kill all of these cells, we really just have to kill the cancer stem cells. The trick is, how do we recognize those cells? So the idea that has emerged here is that over the past 30, 40 years, we've not understanding these differences among cancer cells, we have screened for new cancer therapies based on their ability to shrink tumors. And an idea, and this has not been proven yet, this is an idea, the idea in the field is that because most of the cells within a tumor are these partially specialized cancer cells that have a limited ability to divide, in screening for drugs that will shrink a tumor, we've inadvertently selected for drugs that kill off these cancer cells with limited proliferative potential. So we have all kinds of drugs that can shrink tumors down to the point where we can no longer see them radiologically. And yet we know, in the context of certain cancers, that 99% of those patients, after we've shrunk their tumors back to the point of no longer being able to detect them, we know that 99% of those patients will go on to die of metastatic disease. The idea is that's because we're leaving the cancer stem cells behind. And that instead of screening for drugs that just shrink a tumor, we should be screening for drugs that kill these cancer stem cells. So that even if we don't shrink the tumor, if we kill off the cancer stem cells, we'll convert a malignancy to what's effectively a benign tumor with a limited ability to proliferate. And the one success that we've had in metastatic solid cancer is with cisplatin compounds in testicular cancer, where this is exactly what they do. And you can leave behind the hair and the teeth and the neurons, and they will almost never come back and kill the patient. So now the challenge is to try to apply these approaches to other cancers where these differences have been less apparent. So now, one cancer where this is particularly important is in the context of leukemia, because what we would really like to do is to develop approaches to kill the leukemic stem cells without harming the normal stem cells. And that's a really difficult thing to do because the leukemic stem cells are using all of the same machinery to proliferate as the normal blood-forming stem cells are. And so it's a hard problem to try to find drugs that will kill the leukemic stem cells without harming the normal blood-forming stem cells. And here's why. There are certain genes, and I don't want to get too technical here, called proto-oncogenes that are growth-promoting genes. These are genes that when a cell turns them on, they cause that cell to divide. So these genes that include kind of a lot of pathways that are well known and frequently implicated in cancer, one of which is a gene called BMI1. I'm only mentioning it because I'm going to mention it briefly later. I'm going to mention this again. All you need to know is that this is one of these growth-promoting genes. And so oftentimes you see this overexpressed in cancer cells, and that's the way that the cancer cells divide out of control. But these proto-oncogenes not only promote the proliferation of cancer cells, they also promote the proliferation of the normal stem cells. Now, these proto-oncogenes are normally balanced against tumor suppressor genes, which inhibit growth. So you have growth-promoting signals and growth-inhibiting signals. And these growth-inhibiting signals not only inhibit cancer cell proliferation, but also inhibit normal stem cell self-renewal. And so under normal conditions in normal tissues, the growth-promoting signals are balanced with the growth-inhibiting signals so that everything stays kind of at steady state. And in cancer, you either amplify these signals or you delete these signals. A couple of genes that are important tumor suppressor genes that I'm going to again mention later and try not to get too technical are INK4A, ARF, and PTEN. These genes are powerful growth-inhibiting signals that are frequently deleted by cancer cells. Cancer cells just eliminate them from the genome and thereby don't have this set of brakes anymore on the process and will divide out of control. So the problem here is if all the same mechanisms are regulating both the normal stem cells and the cancer stem cells, how do we kill the cancer stem cells without killing the normal stem cells? In an ideal world, what you'd like is to identify a rare example of a growth-promoting signal or a growth-inhibiting signal that has opposite effects on the normal stem cells and the cancer stem cells. Because if you could do that, then you could target this mechanism to try to kill one without killing the other. And so my lab has identified an example of that in blood-forming stem cells. So blood-forming stem cells, as I've indicated, are present in blood-forming tissues like your bone marrow throughout life, where they make all the types of cells that are present in your blood. The lymphocytes that fight infection, the myeloid cells that gobble up bacteria, megakaryocytes that make the platelets that allow your blood to clot, the red blood cells that allow you to transport oxygen around your body. The stem cells themselves are very rare, but they divide, they give rise to so many dividing progeny that they make literally billions of new blood cells every single day in your bone marrow. Now this illustrates the enormous proliferative capacity of blood-forming stem cells. Here, this is an example of an experiment from my lab where we've taken a single blood-forming stem cell from a mouse and deposited it in a dish and allowed it to divide for 2 weeks. So this is a cloud of blood cells that we've obtained from a single blood-forming stem cell. Each of these little dots is a single cell. And so you're looking at thousands and thousands of blood cells that have arisen from the stem cells. The colony has a kind of reddish appearance because we're actually making red blood cells in the dish. And there's different kinds of cells in these cultures. There are these big clear cells that are probably megakaryocytes, other kinds of macrophages, red blood cells, etc. within this colony. So cancer cells, it turns out, hijack the mechanisms that these normal blood-forming stem cells use to make progeny in a regulated manner to proliferate out of control. And one of the genes that is very consistently, the growth-inhibiting gene, that is very consistently deleted or otherwise inactivated by cancer cells is this gene called PTEN, which is normally a growth-inhibitory signal. Now, we can, with genetic engineering tricks, cause mice to inactivate this PTEN gene. And when we do that, the mice very quickly develop leukemias, just like patients develop cancers when they have mutations that inactivate PTEN in their tissues. And so here we're looking at sections through a spleen and the thymus of a mouse where the spleen is filling up with myeloid leukemia cells, and the thymus of the mouse is filling up with lymphoid leukemia cells, and the spleen increases to 10 times its normal size, just like would happen in a patient with some forms of leukemia. The interesting thing is here, that when we saw that taking PTEN away caused these mice to develop leukemia, we expected that the normal stem cells would also expand in number. But in fact, the normal stem cells have the opposite response. The normal stem cells become depleted after you take PTEN away. So this was a rare example of a machinery that was used differently by the normal stem cells and by the cancer stem cells, and that gave us an entree to think about targeting this pathway with drugs. Because if we could inhibit what was happening as a result of losing PTEN, then we might be able to kill the cancer cells without harming the normal stem cells. And so it turns out there's a drug that's FDA approved for other reasons called rapamycin. And rapamycin counteracts the effects of taking PTEN away. So we treated the mice with rapamycin to see whether, number one, we could treat their leukemias effectively, and number two, rescue their normal stem cell function. And in fact, we could do both things. So from both of these mice, we've inactivated this PTEN gene, and in this case, we didn't treat the mice with anything, and these mice went on to die of leukemia within a month of losing their PTEN gene. Whereas the mice that we treated with rapamycin actually remained happy and healthy, walking around like nothing ever happened. And as long as we kept these mice on rapamycin, they remained perfectly healthy. So at least in the context of mice now, we have the world's first example of a chemotherapy that makes the normal stem cells healthier while it's killing the cancer cells. And so we're hoping, the idea here is that by developing a sophisticated understanding of the mechanisms by which stem cells self-renew, we might be able to systematically identify these differences between normal stem cells and cancer cells to identify new ways of killing cancer that might be more effective at killing the cancer and less toxic to the normal tissues. These results were so unexpected and so promising that this work was published last spring on the cover of a journal called Nature, and in collaboration with our colleagues at the University of Michigan Cancer Center, we're actually planning to go into clinical trials with rapamycin, giving rapamycin on top of existing treatments to certain types of leukemia patients to see if we can cure patients that aren't currently cured. And this is a cool story because we started out with a very basic science question in mouse and had observations, some of which weren't unexpected, some of which were unexpected, that led us to get insights into how rapamycin might be used more effectively against cancer. Now, We don't know whether this is going to work yet. We're going to do a clinical trial, and maybe it won't work, or maybe there'll be people that are cured who wouldn't be cured otherwise. But this is the cool thing about stem cell research. This is why people want it to move forward, because it creates opportunities that don't exist otherwise. And even if it doesn't work in all circumstances, the opportunity to do something is a powerful thing. And if this works some of the time, if we could cure 20% of patients in our study who wouldn't be cured otherwise, that changes the world for a lot of people.
Excuse me, did you make the mouse better, or did you just prevent it from getting sick? Which, with the rapamycin?
Both, if we give it in the right circumstance. If we give it immediately after taking the P10 away, we can prevent that mouse from ever getting sick.
Okay, now that's preventing, but what about healing?
If we waited till the leukemias were raging and then gave the rapamycin, we didn't cure any mice, but we lengthened their lifespans in all cases. So it was still effective, but didn't give a cure. And that has actually influenced the way that we've been thinking about designing the clinical trial at the Cancer Center.
Thank you.
Is it the AML mice have the translocation, the 821? What were your findings on that?
So oftentimes after you take away these tumor suppressor genes, other mutations will also occur, like translocations, that will be growth promoting. Oftentimes cancers have multiple genetic defects that contribute. There probably are translocations involved in this, but we haven't identified exactly which. Okay, so let me tell you a short story about aging. We all know that fundamental to aging is reduced growth, reduced regenerative capacity, increased incidence of degenerative disease, and increased incidence of cancer. Here's a cartoon from the New Yorker of this little old man with a rather oversized walker, and the caption from the ladies in the background is he used to own a muscle car. So if you think about what cells are most responsible for growth and regeneration, and what cells are most likely to be transformed to cancer, we think about stem cells in most cases. And so it raises the possibility that the changes that happen during aging are changes that, at least to a considerable extent, occur at the level of stem cells. And so that's another major thing that my laboratory has been studying. So in this case, just partly to illustrate another kind of stem cell, and the things that I tell you about here are broadly true of stem cells in many different tissues. There's a stem cell population that's present in the forebrain throughout adult life. And the job of these stem cells is to form new neurons that migrate into the olfactory bulb, which is the part of the brain that processes smell. And the new neurons that are born in the olfactory bulb probably regulate your ability to learn to discriminate new odors as an adult. So you all know if you walked into a perfume factory tomorrow, you might have a hard job distinguishing the different types of perfume or remembering what the differences were. Whereas somebody who would, if you worked in that perfume factory for 6 months, then you'd be shocked that it was ever a time when you couldn't tell the difference between them because they would become so familiar to you. That's because we learn to discriminate new odors during adulthood. And it probably involves these stem cells and the new neurons that they make. The problem is that the frequency of new neurons that they make declines dramatically with age. And we've known this for a long time. So in the same way as your muscles become less regenerative and it takes longer to repair injuries, it takes longer for cuts to heal, your brain also generates fewer new neurons as you get older. The problem is nobody knows why. We all know that this is a fundamental characteristic of aging, but we've had no mechanistic insight into why it is that old tissues have less regenerative capacity. We sort of all just think that, well, what do you expect? It's getting worn out with age. Of course it's not going to work as well. It turns out, for better and for worse, it's more complicated and more interesting than that. So I don't know if it's possible to turn the lights down a little bit in the front. I better not start hitting things because it's hard for you to see. But so this is, no, I hit the wrong button. So this is a cross-section through the forebrain of a mouse where these stem cells live. And we've given this mouse a chemical that marks dividing cells. So that lights up the stem cells in these green dots. So each of these little blue dots is a different cell. This is the region of the forebrain where the stem cells live, this bright blue area where the cells are densely packed. Each of these green dots is a dividing progenitor cell that's in the process of making a new neuron. So this is a young adult mouse brain. This is an old adult mouse brain, where now this region where the stem cells live has thinned because there's a lot fewer dividing cells. And whereas the lateral ventricle is a virtual space, that is the fluid-filled cavity along which the stem cells live, is very small in the young adult mouse brain, it becomes huge in the old adult mouse brain because old mice, like old humans, develop cortical atrophy. So for all of you that are over age 35, your brains are shrinking as we speak. And it's partly because there's a decline in the function of these stem cells. And so the question is, why is it that these stem cells are less active with age? We discovered that there's this tumor suppressor gene called INK4A that gets ramped up with age. You can't detect its expression in young adult mice. You can begin to detect it in middle-aged mice, and it dramatically increases its expression level in old adult mice. So this is a gene that's associated with, quote, cellular senescence. That is, in many cell types, when this gene turns on, the cell shuts down. So the fact that this was turning on in stem cells with age made us wonder if this was the reason why there was less stem cell function and less formation of new neurons in the aging adult brain. So to test that, we aged mice that are lacking this gene, in which the gene has been inactivated. And what did we see? Well, in fact, we could partially rescue this age-related decline in stem cell function, and we could increase the rate at which new neurons were formed in the old adult brain. So here, these are again sections through the forebrain of the mouse, where we've administered this chemical that we can use to light up the newly born neurons. So these are newly born neurons in the normal old adult mouse brain. And here's the old adult mouse brain without this INK4A gene. You can see there's just a lot more newborn cells. There's a lot more green dots. If you count, you can almost double the rate of neurogenesis. You can almost double the rate at which new neurons are born. So eliminating this gene has no effect on the rate at which neurons are born in the young adult mouse brain, but you can double the rate at which they're born in the old adult mouse brain. This is important because this was the first time that anyone had shown that they could partially reverse the effects of aging on stem cells or on the formation of neurons by deleting a single gene. And this tells us that things aren't just wearing out with age, because if they were just wearing out, then taking away another gene shouldn't make things better. In fact, stem cells are actively shutting themselves down when we get old by turning on growth-inhibiting genes like this one. Now, without trying to get too technical here, remember how I told you earlier, in normal stem cells, you have a balance between growth-promoting genes, like the BMI1 proto-oncogene, and these growth-inhibiting genes, tumor suppressors like INK4A. These things regulate normal stem cell renewal. They also regulate cancer cell proliferation to the extent that cancers amplify these signals or delete these signals. The data that I've just showed you adds one more piece to the picture. That is, they also regulate stem cell aging. And this makes a lot of sense because we are all evolutionarily selected for maintaining the regenerative capacity of our tissues throughout adult life, while keeping a lid on that regenerative capacity so things don't turn into cancer. And what these data add to that kind of intuitive picture is that the way in which we balance these signals changes with age. That during fetal development, these growth-promoting signals are dominant, and many of these tumor suppressor signals aren't even competent to come on. Because during fetal development, you don't need any breaks. Everything is dividing as fast as it can to create the tissues, and there's no time to accumulate the genetic defects that lead to cancer. Whereas so the growth-promoting signals are important, and the growth-inhibiting signals are not so important during fetal development. As you move into young adulthood, these signals come into balance, where now most stem cells divide only intermittently, but they retain the right to go into cycle in response to injury to repair your tissues. That balance changes again when you get old. Now the growth-inhibiting tumor suppressor signals get ramped up. The good news is that that allows us to get older before we get cancer. the cost, even though you could say that these INK4A inactivated mice aged more slowly to the extent that their tissues stayed younger longer, but the cost was they started developing cancer at 1 year of age. So the good news is that by ramping up these tumor suppressors when we get old, this allows us to get older before we get cancer. The bad news is that it makes us get older. So this work was actually covered by the New York Times, and similar experiments were done in 3 different laboratories in different places at Harvard, and Ned Sharpless, who did similar experiments in the pancreas at the University of North Carolina, was quoted in the New York Times article as saying, "There's no free lunch. You're either going to die of cancer or you're going to die of old age, but we're all doomed." So on that happy note, at the time, we and our colleagues had shown that many different tissues had showed the same kind of response to INK4A as I described here. Many different tissues, the stem cells got shut down with age. About a year later, 5 different groups were looking for genetic variations in humans that were linked to age-related diseases. That is, genetic variations you could find in the general population that were linked to higher risk of type 2 diabetes, coronary heart disease, or myocardial infarction. The only gene that was found in all 5 studies was this INK4A gene that we had previously found in the mouse. And so it raises the possibility that this gene is very broadly involved in the diseases of aging. And it raises the possibility that if we had drugs to be able to inhibit the function of the gene product that's encoded by this gene, we might be able to use those drugs to promote regenerative capacity in aging tissues. And that's, again, a kind of idea that we wouldn't have thought of in the absence of this stem cell research. Okay. So let's talk a little bit about embryonic stem cells. I've given you some examples of adult stem cells and the great things that we're learning from adult stem cells and why it's important. When I transition to embryonic stem cells, I feel a little bit like this guy whose boss is saying to him, "I'd like your honest, unbiased, and possibly career-ending opinion on something." So embryonic stem cells, as I indicated earlier, only exist at the very earliest stages of embryonic development, and they can form any cell type in the body. Now, human embryonic stem cells were first derived by James Thompson at the University of Wisconsin back in 1998. So we have had only a limited number of years to be able to study embryonic stem cells. In biomedical research, it typically takes us 10, 20, 30 years to go through the various phases of research before you actually get to something that can actually help a patient. So these are early days with respect to embryonic stem cells. Now, why are embryonic stem cells important? They're important for 3 general ways. First of all, in terms of cell therapy, this is the replacement parts idea. There are certain diseases like Parkinson's disease and juvenile diabetes that are caused by the loss of a certain specific cell type, and where we know that those disease processes can be reversed just by replacing that cell type. To the extent that embryonic stem cells can make any cell type in the body in unlimited quantities, this is really a dream come true with respect to being able to cure a major public health problem like juvenile diabetes or Parkinson's disease. Secondly, it offers the opportunity to model human inherited diseases in a way that wasn't possible before. So we've known for many years, for example, about genetic defects that cause a greatly increased risk of early onset Alzheimer's disease. And what we've historically done in the medical research community is as soon as we identify a genetic defect in patients that causes disease, the first thing that we do is we make mice that carry the same genetic defect so we can study the disease in mice. The problem is that sometimes the disease in mice doesn't look like the disease in patients. And Alzheimer's disease is arguably one unfortunate example of this, where despite having identified a genetic defect that's responsible for Alzheimer's disease, there is still great controversy about how that leads to the disease because the mice don't develop a disease that looks like the human disease. In contrast, with embryonic stem cells now, we have the possibility of taking the exact genetic defects that cause disease in patients and putting those genetic defects in embryonic stem cells, and then using the ability of embryonic stem cells to make whatever cell type is affected in that disease in a dish. So in the case of Alzheimer's disease, we can make human brain cells in a dish that have Alzheimer's disease. And we can watch in real time what goes wrong first, what goes wrong second, and hopefully better understand how the disease occurs. But not only that, we can then exploit the ability of embryonic stem cells to make unlimited quantities of the cells we're interested in to discover safer and more effective new drugs. And this is a kind of application that is absolutely not possible otherwise, because where are we going to get human brain cells with Alzheimer's disease? It's not like anybody has any to spare that they can give up. And even if you try to biopsy brains from patients after they die, you don't get the kind of dividing cells that you need to generate the numbers of cells that are required for drug screening. If you go to Novartis where they screen for new drugs, they literally have robots that are around the clock screening through millions of potential drug candidates in tiny little wells. And when they do a screen like that, they need to have cells that can make unlimited quantities of cells. Embryonic stem cells can do that in a way that has not been possible previously. And this is why Nancy Reagan talks about the importance of embryonic stem cell research to the future of Alzheimer's disease. So how does it all work? Well, the embryonic stem cell lines are grown in unspecialized form in a dish. And then we can take the chemicals that are normally responsible, that normally cause particular tissues to develop in the body, and put them in the dish to cause the embryonic stem cells to become specialized into particular types of derivatives. So we can give them certain signals that would cause them to turn into blood-forming cells that we could transplant into a child that's been treated for leukemia. Other signals would cause them to turn into brain cells that might be transplanted into a Parkinson's patient. Other signals might turn them into heart muscle cells that might one day be transplanted into patients after a heart attack. Other signals would cause them to differentiate into insulin-secreting cells that could be transplanted into a patient with juvenile diabetes. So embryonic stem cells can do two kinds of things that adult stem cells can't do. First of all, they can make unlimited quantities of cells, whereas adult stem cells can make limited numbers, large numbers, but ultimately limited numbers of progeny. This has been a problem, for example, in clinical trials for Parkinson's disease, where aborted human fetal tissue was used to try to replace the dopamine-secreting neurons that go missing in the brains of patients with Parkinson's disease. And there's just not enough of that tissue to treat all of the patients that could have potentially been treated with that. And so when you're dealing with a major public health problem, the ability to make unlimited numbers of cells is not insignificant. The second thing, as I've already mentioned, is that embryonic stem cells can make all cell types in the body, unlike adult stem cells that are partially specialized. Umbilical cord blood cells are really good at making new blood cells, but they're not good at making cells in other tissues. So they're great for replacing the blood-forming system in children, but they're not very good for treating other diseases. And that's important because, for example, in the pancreas, the organ that's affected by juvenile diabetes, as far as anyone can tell so far, there is no adult stem cell. And so the Juvenile Diabetes Research Foundation, an organization created by parents whose children are affected by juvenile diabetes, get very frustrated when opponents of this work talk about how we can do all the same things with adult stem cells. 95% of my laboratory focuses on adult stem cells, and I can tell you it would be great for us if we could do everything we wanted with adult stem cells, but we can't. There are certain things that embryonic stem cells can do, like making insulin-secreting cells, that adult stem cells can't do. And this is why the Juvenile Diabetes Research Foundation believes that embryonic stem cells represent the best hope for a cure to their children, and I think is at the top of the list of major public health problems where it's reasonable to think that we may be able to improve the treatment using embryonic stem cells. So the point here is that if we're really serious about curing disease, we should be using all of the weapons at our disposal. And research should be proceeding both with adult stem cells and with embryonic stem cells. And I'll come back to that point later. So you hear many different people pointing to many different diseases that they suggest may be effectively treated with embryonic stem cells. The fact is that it's early days, and there's much more research that needs to be done, and we don't really know at this point what things we're going to be able to treat more effectively, which things might benefit more from adult stem cells, which things might benefit more from embryonic stem cells. But unless we do the research, we're never going to find out. And that's why it's so important to get the research moving now with all of the kinds of cells at our disposal, because if there's one thing that we know from our history of studying stem cells, is that we've been very bad historically at making predictions about which cells are best for which jobs. So let me focus a little bit more on juvenile diabetes, just for a moment. It's caused by the loss of insulin-secreting cells in the pancreas. We know from a clinical procedure called the Edmonton Protocol that juvenile diabetes can be effectively treated by replacing those insulin-secreting cells. Right now, doctors can extract insulin-secreting cells from the pancreases of cadavers and transplant them into kids with juvenile diabetes and cause those kids to no longer need to inject themselves with insulin. So this is proof of principle. The problem is that there's not enough qualified donors out there to be able to transplant all the patients that could potentially benefit. So what we really need is some unlimited source of cells that can make insulin-secreting cells. Enter embryonic stem cells. And this is why the Juvenile Diabetes Research Foundation has so focused on embryonic stem cell research and has led the way in many ways in terms of public policy to try to loosen restrictions on embryonic stem cell research. So where do embryonic stem cells come from? Well, couples that have fertility problems will enlist the help of a fertility clinic where sperm and egg are mixed together in a dish to form embryos. Now, whether you get pregnant the old-fashioned way or whether you do it by in vitro fertilization, not every embryo develops normally, and not every embryo can form a pregnancy. And so as a result, what the doctors do is they want to watch these embryos develop in the dish for a few days to figure out which ones are healthy and which ones are not, so that they can implant the healthy ones in the mother and not implant the unhealthy ones. And as they go through that process, on average, there's about a third of the embryos that look really healthy. Those are the grade A embryos that get implanted in the mother. There's another group of embryos which look healthy enough, but are not as good looking as the other ones. Those are often frozen down in case the first embryos don't take and the parents want to come back and try again. There's also about a third of the embryos which don't develop in a healthy manner at all, and are either known to carry very serious genetic defects that cause diseases incompatible with life, or that become completely arrested and fail to develop, for example, in the dish. Those embryos are immediately thrown away. And whether you're talking about the embryos that don't develop in a way that's healthy enough for implantation into a mother, or whether you're talking about the embryos that get frozen down, we could use those embryos to derive new embryonic stem cell lines that would enhance our ability to study human disease. Now, when I say embryo, one of the difficult things that happens is that people get a picture in their mind of something that looks vaguely like a baby. You know, something that looks like what you'd see on an anti-abortion poster. What I'm talking about has nothing to do with abortion. Abortion is after you take an embryo or a fetus that has developed to a certain point after implantation in somebody's uterus and remove it. I'm talking about dealing with embryos within a few days of fertilization that have never been implanted in anybody's uterus. So this is a picture of what these blastocysts look at this stage of development. These are microscopic balls of cells with no specialized tissues. There's no heart, there's no nervous system, there's only a few hundred cells. And it's these blastocysts which are routinely discarded for all kinds of reasons from fertility clinics, and which could be used to enhance embryonic stem cell research. Now, thousands of embryos are routinely discarded by fertility clinics. These embryos could never be used for fertility treatment, either because the parents make the decision that they're going to discard those embryos, or because they're not developing in a healthy enough manner for the doctors to be willing to implant them. Even though they could not be used for fertility treatment, they could be used to develop new human embryonic stem cell lines. There's broad agreement on how this can be done ethically. The agency that advises our federal government on matters of science and technology policy is the National Academy of Sciences. They studied this issue about 2 years ago, and they found that embryonic stem cell research was critical for the future of medical research in the United States, and that they recommended specific guidelines by which embryonic stem cell lines could be safely and ethically derived, and those guidelines have been broadly adopted by universities and research institutions across the country. So the fact is that among the people that really know about this work, there is very broad agreement on how it can be done ethically. Most recently, Dr. Zerhouni, who is the director of our National Institutes of Health and who was appointed to that post by President Bush, testified before Congress saying, "From my standpoint, it is clear today that American science will be better served and the nation will be better served if we let our scientists have access to more embryonic stem cell lines." Now, the opponents of this work often act as though we're making a choice between doing medical research or making babies. And how could you contemplate taking these embryos that could otherwise be used to make babies and doing medical research? It's absolutely not true. They're trying to reframe the debate in a way that's not accurate. President Bush frequently presents this misinformation when he talks about this subject. The decision before us is not to use embryos to make babies versus using them to make embryonic stem cell lines. The decision is just, with respect to the fraction of embryos that are already being thrown away by fertility clinics and could never be used for fertility treatment, the decision is, do we just throw them away, or do we take those embryos and try to use them in medical research that might one day help patients? The fact is that even though the federal restrictions are defended by the president and the restrictions in certain states are defended by state legislatures as being necessary to protect human embryos, in fact, those restrictions don't prevent the destruction of a single human embryo because it's perfectly legal to throw these things away. These restrictions only delay medical research, and it's one of the reasons why proponents of this work get so frustrated. Okay, so I want to make one other point, and then I'll wrap up. And that is, you also hear the debate about nuclear transfer. And what's nuclear transfer? Well, if you just take cells from my body and try to transplant it into one of your bodies, your immune system will recognize my cells as being foreign and reject them. And so we need to have a way of getting around that in order to effectively deliver the benefits of this research to a diverse patient population. And so nuclear transfer is the way that scientists have proposed to do this. So this is therapeutic cloning, where what in fact happens is that you can take the genetic material out of a human egg, or out of an egg, I should say. This has been shown to work in a variety of animal species, but has not yet been shown to work in human cells. So this was the big fraud that was perpetrated in Korea, where the Koreans claimed to have done this successfully. In fact, it turned out that they hadn't. Now, Harvard University and the University of California at San Francisco have announced major programs to try to do this for real. So the idea is that if we were trying to transplant cells into Michael's body to cure him of something, we could take a punch biopsy from his skin, grow out his skin cells, take the genetic material out of his skin cell, and inject it into a human egg from which the genetic material has been taken away. Now, these eggs never get fertilized. There's no sperm anywhere involved in this process. But we can trick these eggs into thinking they've been fertilized so that they start to divide. Now, they don't develop normally. Even if you transplanted them into somebody's uterus, they rarely, if ever, would be able to make a pregnancy because they don't develop normally. But in the dish, the cells that form in the center of these blastocysts can be taken out and grown into an embryonic stem cell line. This has worked in a variety of animal species. Scientists would like to be able to make it work in humans. Because the great benefit here is that now, because these embryonic stem cells carry Michael's genetic material, they are genetically identical to him. And once we form cells from these, they can be transplanted in his body, and his immune system will not reject them. So this is the other part of the equation that scientists talk about wanting to do. This is perfectly legal under federal law, but in certain states, such as the state that I'm from, it's illegal to transfer genetic material into a human egg. Now, the argument against this is that people say, well, what if scientists took these things and transplanted them into somebody's uterus? they could be used for reproductive cloning. And it's true, we don't want to do reproductive cloning because what we've learned from trying it in animals is that it's unsafe. It doesn't work 99.99% of the time, so no one should try it. Everyone agrees no one should do that in humans. But if we want the benefits of this research, we should make it illegal to transfer these cells into a uterus, because as long as they never go into a uterus, they can never create a pregnancy, rather than just making it illegal to transfer genetic material into a human egg to begin with, which prevents us from doing therapeutic cloning that could potentially benefit patients. Now, of course, the trick is here, as is illustrated in this cartoon, the trick is to separate the nuclei, because you have these interlocking spheres of religion and politics that come into play here with many different points of view. So why would additional embryonic stem cell lines help? Well, the federally approved lines that we have available for federally funded scientists are all contaminated with animal products, and that may be a real problem for using these lines in patients. We may be able to better understand the origin of disease with new lines, because right now, none of the federally approved lines allow us to model any specific human diseases, whereas newer lines that are being created carry the kinds of genetic defects that cause disease. Finally, improved drug development would come from having more lines to reflect a more diverse patient population. When I apply to the National Institutes of Health for a grant to do clinical trials, I have to tell them how many African Americans, how many Asians, how many Caucasians are going to be entered in the trial, how many men, how many women. Because if we only try new drugs on white males, there's a chance that we'll develop medicines that only work on white males, and that's not what we want. And yet, the same federal government forces us to only use embryonic stem cell lines that come from overwhelmingly a single fertility clinic in Haifa, Israel, that don't even come close to representing the genetic diversity in our society. It's possible that none of the federally approved embryonic stem cell lines come from an African American background, for example. If we could derive new lines and use them in a federally funded lab, we could solve that problem. So I'm going to leave you with 2 points very briefly. First is to remind you again that current federal and state restrictions do not protect a single embryo from destruction. They only delay medical research. The embryos that we would use for this process are already being thrown away and could never be used for fertility treatment. And secondly, the framing of this debate as, are adult stem cells better, are embryonic stem cells better, this is a political artifact. This makes no sense to stem cell biologists. I've never met a stem cell biologist who believed embryonic stem cells were better or adult stem cells were better. It's a bit like wanting to debate whether carpenters should use hammers or screwdrivers, right? It would make no sense to a carpenter. Of course you need both because they do different jobs. Well, every stem cell biologist that I know believes we should be working on both adult and embryonic stem cells because we don't know which ones are going to work best in which context. So this is not a contest between adult versus embryonic stem cells. This is a contest between us and disease, where we really should be using all of the weapons at our disposal, and we're currently fighting with one arm tied behind our back as a result of the restrictions that are on embryonic stem cells. So I apologize for going a little bit long, and I appreciate your attention. I'd be happy to take any questions that you have.
Well, Sean, thank you very much for this outstanding and captivating talk. If you have questions, please raise your hand. We will bring you a microphone so that everybody can hear. I also ask you to fill out the surveys so that we have feedback on our program. And who wants to take the first question, please?
Hi, I didn't hear any mention of mitochondrial DNA or any of the other cytoplasmic components in the oocyte. What effect are we talking about here? Thank you.
Yeah, one of the challenges to me is that this is a heterogeneous audience where some people have a limited biology background and some people are really expert, and I tried to pitch it in the middle. So the question is about mitochondrial DNA, and the idea here is that apart from your genetic material, in the nucleus, which is the main genetic material that determines your characteristics, hair color, etc., there's also genetic material in mitochondria, which is a subcellular component that generates energy for the cells. And when you do therapeutic cloning, you're replacing the nuclear genetic material without affecting that mitochondrial genetic material. And this has been a theoretical concern in the field. Will the genetic material in the mitochondria sort of mess up the process? And I think the experience so far from cloning in animals is that it doesn't get in the way, and so it's not a practical problem. There's a question here in the middle. There's a question right at the back.
So I can understand why researchers would want to use embryos that are going to be discarded from fertility clinics, but when you want to get the ones that are non-viable or are genetically mutated and just won't give rise to life, how much are you going to learn from those because they're not giving rise to normal embryonic stem cells?
So those are some of the best ones to study because those oftentimes carry diseases that we would like to learn more about. So let me give you an example. There are communities in Brazil in which Huntington's disease is endemic, where there's a particular genetic defect where if you inherit that genetic defect, you're going to develop Huntington's disease. And so the way that they've started to deal with this is affected families go to fertility clinics and they can genetically test which embryos have the Huntington's disease gene and which ones don't. And their practice is to implant the healthy ones and to throw away 100% of the embryos that are affected by Huntington's disease. Now, if we could take those embryos with Huntington's disease in my laboratory and make embryonic stem cell lines, maybe we can better understand how Huntington's disease is caused and identify drugs that would cure Huntington's disease. And then it would no longer be necessary for these families to throw these embryos away. And so that's an example of a context where even though these embryos would normally be thrown away, we could use them to better understand disease. And this is why people like Orrin Hatch with very strong pro-life voting records describe embryonic stem cell research as the pro-life position. In the back?
How did the existing stem cell lines become contaminated? Can you explain that?
Yeah, particularly in the early days of growing these lines, they had to be grown on top of what are called feeder cells. That is, other cells had to be around to secrete growth factors to keep the stem cells alive. And in the early days, these feeder cells were all derived from mice. So all of the federally approved lines were grown on mouse cells. Now, our own Food and Drug Administration is loath to allow people to put into patients any kind of product that's contaminated with animal products because there's the possibility of transmitting viruses from animals into patients, and there's also certain kinds of sugar structures that are made by animals that are not made by humans and that could be attacked by a human immune system. And so as a result of those changes, the field has long wanted to find a way of growing these cells without animal feeders. That has been accomplished in recent years, and so many of the newer lines are not contaminated by animal products. But under the president's policy, federally funded researchers in this country are not allowed to study those lines. Down here is a question.
So what's happening in the rest of the world?
So the question is, what's happening in the rest of the world? Well, in the same way as the states in the United States represent a patchwork of different kinds of legal situations, you see the same thing when you look across the world. There are many countries like Great Britain and Australia and China and Singapore and Japan that have more liberal laws than are existing in the United States and much more government support for this type of work. There are also other countries on the other end of the spectrum, like Germany, which have more restrictive laws. Canada, for example, is more liberal in some ways and more restrictive in other ways. So the difficult thing is that America has been the world's engine for biomedical research. America is the country where most things get discovered first. And so when the science is slowed in America, it really does affect the pace at which things develop in the world.
What's the current status of research on dedifferentiation of adult stem cells back to embryonic stem cells?
There's been really exciting developments just in the last 2 years in that area, where a couple of research teams have shockingly identified a group of just 4 proteins that, or 4 genes, that when put into skin cells grown from mice, can turn those skin cells into embryonic stem cells. And this is really exciting because this gives us a cool way of making potentially new embryonic stem cell lines that doesn't involve the destruction of an embryo. So scientists are very interested in this for a variety of reasons, but there's a couple of problems that mean that this does not replace the need to use new embryonic stem cell lines that we have or to derive new lines by destroying embryos. First, none of these techniques have worked in human cells yet, and it remains to be seen. I think it will ultimately work with human cells, but we don't know how long it'll take. The second problem is that the way that the scientists put these genes in mutates the cells. And they already know that it causes these cells to sometimes turn into cancer cells. And you wouldn't want to be treating patients with cells that you were worried were going to turn into cancer cells. And so it will never completely replace, I think, the need to derive embryonic stem cell lines from embryos, but for certain kinds of applications it may be desirable to do the dedifferentiation approach once it's possible with human cells. There's one in the back, one here.
Actually, this is a bit more biological question. What does rapamycin do? How does it work?
So rapamycin inhibits mTOR, the mammalian target of rapamycin, which is a kinase that's downstream of PTEN in the signaling pathway that PTEN's part of. So when you take PTEN away, there's a particular signal in cells that gets dramatically amplified. And one of the things that it activates is this protein called mTOR. Rapamycin reverses that activation. And it's been used as an immunosuppressant because this protein is involved in immune responses, but it turns out it can also be an anti-cancer agent if used in the right context. Back?
Has there been any progress with privately funded stem cell research?
What exactly do you mean by progress?
Do they have like newer stem cell lines for which federal funding is not available?
Yeah, absolutely. So my employer is actually the Howard Hughes Medical Institute. It's one of the biggest philanthropies in the United States. It has $14 billion, so it spends hundreds of millions of dollars every year supporting medical research. One of the people they support is Doug Melton, who is the co-director of the Harvard Stem Cell Institutes. And his laboratory with Howard Hughes funding has actually used that private funding to make more new human embryonic stem cell lines than were created with all of the public sources of funding put together prior to his work. The problem, and he is willing to give those lines out free of charge to anyone who wants them. The problem is that most scientists in this country, including most scientists at Northwestern University, depend on federal funds to run their laboratories, and therefore most laboratories have little or no ability to use those newer lines that Doug has developed. Right here.
If you're going to use stem cells to replace damaged or non-existent pancreatic beta cells or whatever, there has to be a way of transplanting them into a recipient, but you also have to have a way of getting them to stop growing when they have grown enough to replace the missing cells. So you need much more than just getting them to grow. You need to control them. Is there any work going on in these growth arresting signals when, I mean, the pancreas knows when it has enough beta cells and it stops making new beta cells?
Yes. So good question. And the way that people deal with this problem when they work with embryonic stem cells is to not transplant the embryonic stem cells themselves. In fact, the embryonic stem cells don't know when to stop. They're like these fetal cells that are dividing like crazy. And so what you do is you differentiate the embryonic stem cells in a dish into the partly specialized cells before you transplant them. So you don't transplant a patient with juvenile diabetes with the embryonic stem cells, you transplant them with the purified insulin-secreting cells that you've developed from the embryonic stem cells. And when you transplant the specialized cells, they then behave like the normal specialized cells in the body and are subject to the normal controls.
Hi, I read about some therapies with adult stem cells with people with spinal cord injuries in Germany and in Ecuador. Do you have any thoughts or information about those?
Well, I don't know, of course, what specific therapies that you're talking about, but one of the things that you have to be careful about is people selling snake oil. Some people with spinal cord injuries have sometimes gone overseas to China or to Eastern Europe, to places like that, to get therapies that weren't being offered in the United States because there wasn't good science supporting those therapies. So I don't know exactly what you're talking about. And it's possible that there are some kinds of therapies that might have certain advantages, but the bottom line is that we can't do much to repair a spinal cord injury with any adult stem cell therapy that's been examined so far. Not to say we might not discover something in the future, it's worth additional research. A company called Geron is going into clinical trials with embryonic stem cells to try to enhance recovery after spinal cord injury. And they have compelling data from animal models showing that they can get rats to walk again. But it's a complicated problem, and it remains to be seen how it'll work in humans. The good news is that they're going into clinical trials. They're trying, and so we have a chance. And it's important to remember here that it's not realistic probably to talk about squirting some cells into a damaged spinal cord and having the person then stand up out of their wheelchair. What the patients with spinal cord injuries say they really want is some recovery of function. It makes all the difference in the world to those patients, even if they could never walk again. If they can regain bladder control, that's huge. So sometimes just regaining an inch of function in the spinal cord will make all the difference in the world. And so the hope is that using cells from embryonic stem cells, or where the evidence is best, at least so far, that maybe we'll be able to partially regenerate function in patients with spinal cord injuries.
When you think about the drug testing of the stem cells, do you think it could be important to have the cells in a three-dimensional structure? Do they react differently to these drugs?
Yeah. Yeah, it's a really good question. In fact, historically, the question is, would cells that are present in a three-dimensional structure, like in our bodies, respond differently to drugs than cells that are growing flat on a dish, which is how we normally study cells in dishes? And in fact, that's true, and there's plenty of evidence to support that. And one of my colleagues at the University of Michigan is proposing to screen for new drugs specifically in cells that are grown in 3 dimensions instead of 2 dimensions for that very reason. My laboratory has been funded by the Department of Defense, for example, to try to create hybrid organs in a dish, 3-dimensional structures to try to mimic the way organs function, to better restore function in soldiers that have had, injuries from gunshots, to have better biological sensors for chemical warfare agents, and to do better drug discovery.
You've talked about how the current political climate fractures the development of science, and then you moved into the offshore capabilities in Singapore, Japan, and other areas. Now, given that you have to take these into clinical trials, how do you see this working out? What are going to be the divisions there? Because if these labs produce something that then goes to a clinical trial, how will the oversight and regulatory environment affect what happens in terms of our acceptance of any new discoveries?
The people in the back hear that? Do you want me to repeat it? Would you want me to repeat it? It's a long question. So I don't know if I can repeat it. So the question was, given that there are different regulatory environments overseas, like in Japan versus in the United States, what will it mean when embryonic stem cell therapies go to clinical trials, and how will those therapies be accepted given the regulatory constraints that may exist within the United States? My personal view is that one of the unfortunate things about this debate is that once it becomes possible to cure a disease with embryonic stem cells, there will no longer be any debate about doing embryonic stem cell research in the United States. That Americans won't stand for a situation where people could be cured of their disease in Japan, but not in the United States because of government restrictions. And that's sad, because what it means is that the restrictions that we have now are just delaying our ability to get there. And I think it's a cynical thing, because the sense I get when I go around Congress is that nobody thinks these restrictions are going to last very long, and they're just sort of trying to hold out against progress. So I think that we'll get there, and a question is how long it'll take. And an important consideration within the United States is this patchwork of different laws within the country. So states like California have explicitly made it legal to do nuclear transfer, to derive new embryonic stem cell lines, and they have invested $3 billion of state taxpayer money in this research, believing that it's so important for the health and economic viability of their citizens going forward. States like Michigan, at the other end of the spectrum, I would go to jail for doing in Michigan what the state of California would give my laboratory millions of dollars to do. Now, are the people of California, are the people in Illinois and Wisconsin, for that matter, who do the same things as people in California can do, are our values in Michigan so different from your values in Illinois that we should go to jail and you should get state government grants? It makes no sense, particularly given that we're not talking about diverting embryos from reproductive purposes. We're only diverting embryos from the trash for this research.
Kind of to follow up on that question, are the political opponents of embryonic stem cell research themselves aware of what you just explained?
My sense is that they are. My sense is that when I go around Congress or when I go around state legislatures, the reaction I get from most people who don't support this research is, oh yeah, we know that you're right, but we just can't do this politically. Now, I haven't discussed it with President Bush, so I can't tell you what he's thinking. But in my experience, these people are not stupid. These people have political reasons for wanting to do what that they want to do. And of course, you know, different people have their own strongly felt moral views. And there are some people in our society who believe that it's wrong to destroy a human embryo for whatever purpose, that it's wrong for fertility clinics to do this in the first place, that it's wrong for fertility clinics to throw these embryos away, and that by extension it would be wrong for us even if we could benefit a patient from using the products of this research. And I respect people that hold that point of view. The problem is that there's not very many people in our society that hold that point of view. And we've known for many years that there are people in our society who believe it's morally wrong to get a blood transfusion. And so they don't have to have a blood transfusion. But because there's a subset of people in our society who believe blood transfusions are wrong, we don't prevent the rest of us from getting blood transfusions when we need one. And so I think that this embryonic stem cell debate is going to evolve much like the in vitro fertilization debates did 30 years ago, where initially the response from opponents, and in fact it's similar groups that opposed in vitro fertilization 30 years ago and who oppose embryonic stem cell research now, who in many ways want to refight the same battle that they lost 30 years ago. 30 years ago they said things like, we're playing God, this hasn't been sufficiently tested, you're turning women into lab rats, you're going to create Frankensteins, you wait, this is going to be a disaster. Of course, none of that was true. It was all fear-mongering in the service of trying to create uncertainty around a new technology to try to detract from public support. People's desire to have children that couldn't have been born otherwise won out in that circumstance, and in vitro fertilization is now a mainstream fertility practice that is widely accepted by most but not everybody in our society. And we don't think about it creating Frankensteins, and the numbers aren't exactly available, but there's probably something on the order of 500,000 children that have been born as a result of in vitro fertilization that would not have been born if that debate had gone the other way. I think that as we see these state initiatives going forward in California and New York and Massachusetts, it's clear that people are speaking And they want this research to go forward as quickly as possible. And whether we're able to cure 1 disease or 10 diseases with the results of embryonic stem cell research, we're going to answer those questions faster if we can do the research now.
Maybe one last question here in the front.
I guess I have a two-part question. The first part is, did I hear you correctly that in California, Illinois, Wisconsin, and then you just added a couple of other states, state law trumps federal law? And then my second part of the question is, what is your objective in doing these talks, I'm guessing around the country, is that correct?
Well, in the first question, there is no federal law against the destruction of human embryos or against nuclear transfer. The federal restrictions are just on how federal grant money can be spent. And so under federal law, somebody in Illinois who has private funding can do whatever they, I mean, they can't do whatever they want. That has to, everything is approved by IRB committees and institutions and escrow committees that specifically oversee anything to do with embryonic stem cells. So there's layers of ethical review that have to occur. But it is legal to derive new embryonic stem cell lines, and it's legal to do nuclear transfer if you can pay for it with some source other than federal funds. That's a problem, though, because most people in this country are dependent on federal funding, and so it slows the process down. And so the states that have gone ahead and usually the federal government leads the way when we're talking about major public health issues. And this is a rare example where that's not happening. And so individual states like Massachusetts and New York and California and New Jersey and Maryland and Connecticut and Illinois and Wisconsin have stepped in where the federal government has failed to go under the Bush administration and said, well, if the federal government is not going to fund this research, we're going to do it. And so that's stimulating that research in those states. But it leaves major universities like the University of Michigan that are actually among the top institutions in the world with respect to adult stem cell research, really unable to devote our full talents to embryonic stem cell research, slowing the enterprise for that reason. Now, with respect to what I do, I go around the world talking about science from my laboratory. I don't really travel around the world giving this kind of talk, but I do this sort of thing periodically like this, and particularly in the Michigan legislature and in Congress, because in my experience, people don't fully understand how the legislative process works. And the fact is that if you don't educate people about these issues, you get bad laws for bad reasons. Because most people, most of the time, don't have an intimate knowledge of what they're voting on in Congress. And it makes a difference to get in front of people and explain what's really at stake. And the Juvenile Diabetes Research Foundation, for example, has done an outstanding job of that federally. And that's what has engendered this broad bipartisan support for loosened restrictions on Capitol Hill. The problem has been that the president has repeatedly vetoed that. But most people believe that almost no matter who wins the next election, that the current president's policies will be null and void come 2009.
Thank you once again for your interest. Thank you, Sean, for your outstanding talk. And please fill out the survey and leave it outside. Have a nice evening.